Started. Okay, thank you. So my name is Barbara Baer, and thank you very much for inviting me. I'm excited to be here, and it's a bit of a special format, so let's get this going. And thanks so much for the nice introduction. I'm starting to get some serious imposter syndrome after this, so I'll try to stay human during the talk. So, research at cyber. Let's go. Would be nice if the slide would move forward. Let me see whether that works. There we go. My accent is from Switzerland. So that's Switzerland, the Vierwaldstätter See. I'm from Zurich. That's where I grew up. And that's where I got most of my accent from. And I met Boris during our PhD in Switzerland when we were working on bumblebees. And since then, we've basically been partners in crime. For 3 years, we have lived in Copenhagen. After the PhD, Boris went on for a postdoc, and we started a family with our first son and lived there for 3 years. Working on insects as well, but this time on leaf-cutting ants. That's a story for another time. After Copenhagen, we moved to Perth in Western Australia and lived there for 13 years. Here you see the harbour along the Swan River in Perth, Western Australia. In Western Australia is a huge state, a huge part of Australia, almost 100,000 square miles for 2.8 million people. And 2.5 million people actually live in Perth. So you can imagine there's a lot of empty space. And these two are my sons here, being at the end of the world. That was one of our last trips before we had a big move. You can see the garage with all our boxes here. And it was a huge move with lots of boxes and far away. So it was a big, big change in 2017 for all of us. And now the bears are in the USA. And it's been a roller coaster, as you can imagine. A lot of excitement, ups and downs as it goes when you move country. And now to the Center for Integrative Bee Research. That's the reason why we are here. I am going to talk to you about the Center for Integrative Bee Research, what it is, our facilities, and hive management we do for our research. The Center for Integrative Bee Research is located at UC Riverside, the University of California, Riverside. And you can see the logo up there. It has just recently changed to this beautiful blue and yellow. And the location is nice. When you are at UC Riverside, there is a whole building just dedicated to insect nerds. And that's of course very exciting when you're an insect nerd yourself. When you come in through the main entrance, you can see here, you can walk up the stairs and you will find our labs right here. This is our lab where we do all kinds of work, and it's very tidy and empty at the moment. And that's a good time to take a picture when everything is tidy and clear. We can do molecular work here. We have microscopes, we have freezers, we have storage space. It's a really, really nice, well-equipped lab. And we have researchers in there sometimes. So this is what we look like when we try to look official for a group photo. We wear lab coats, and here we are at the insemination workshop, and we have people with lots of different expertises here. So let's start with our engineer, a molecular biologist, a professor, someone who learns beekeeping, who teaches us beekeeping and learns beekeeping with us, and AI, Owen and Jessica, grad students and a master student. So yeah, you see there is a lot of different people working here. This is not our whole team. This is an older picture, but yes, the teams change a lot. We are here in Riverside at UC Riverside to develop beekeeping tools. That's basically what keeps us going. And the reason we want to develop beekeeping tools is that we would like to improve beekeeping in general, make it easier on people, make it easier and better on the bees. Because when there are 10 beekeepers, there are 15 opinions, and that's a good thing because then we are innovative everywhere. So if we have as many different new tools as possible, it will give us a chance to try them all and see what works best for everybody. We can pick and choose the combinations that work best for the best way to beekeep. To do this, we are integrating scientific methods. For example, experimental ecology, and I'll give you an example of that. Molecular biology, where we study the molecules and the genes, and engineering. These are the three main scientific fields we are employing. And we study different parts of, we do bee research mainly on the bee reproduction, so their immune system and the environment, hive environment and outside environment of the beehives. We do this to develop those beekeeping tools I've been talking to you about, and I'm going to give you an example like that. So one example for experimental ecology, how we integrate it with the immune system of the bee, is the gut parasite Nosema. And Nosema causes diarrhea. This is a hive that has a heavy infection with or bees with lots of diarrhea. The Nosema parasite is a spore, a microsporidian. So it's a one cellular organism living in the gut of the bee. Here you see a bee gut, and this is the midgut. This is where that Nosema parasite is located. And a big part of when you're working with that parasite is that you dissect out these guts, isolate the spores, and then feed them back to bees. In this case, it's a drone. I did some research on drones and that parasite, but we do it on workers as well. And then you count how many spores they keep in their guts and how long they live, and whether there are different reactions, whether different colonies have different reactions to this parasite, whether some colonies are more resistant or doing better with this parasite than others. You investigate these bees when they are infected with 10,000 spores each. How long do they live in a cage? And how infected are their guts after a week or two? We also study the immune system of the bees, or the hemolymph of the bees. And this is a molecular gel. See here, the method has changed. We're going down to the molecules in the blood of bees, and each of these dots is a protein. The proteins are distributed along an electrical gradient and along a distance gradient, and you can isolate these different proteins. And by using methods like these, we have already found 4 candidates for a bee-derived medication that actually kills this Nosema apispore efficiently. And there are 2 grad students following up on this research, actually, yes, and to find this bee-derived medication. It's quite exciting, and we're keeping that. We started the work in Australia, and we keep on working on this here in the US. This makes that wheel for CYBER, the Center for Integrated Bee Research. So that's what integrated bee research means to us. We use the methods of experimental ecology, molecular biology, engineering to study the reproduction, the immune system, and the environment of our honeybees. And then we develop things like a bee-derived medication. We want to develop a bee breeding program and hive sensors from all of this. Those are the main tools, but underneath them, of course, you have different smaller sub-tools that we will develop as we go. And we would love to work together with you to develop these things, especially with the breeding program. We've already attempted work, and the hive sensors. This is an example for a hive sensor, how it is placed in the heart of the hive. And this is Danny, who is testing it out, how the bees are reacting to it, whether they are gluing it in with wax and so on. and doing some preliminary work to find out how the sensor can send data to the cloud, and from the cloud to mobile phones to alarm beekeepers, hey, there's something going on, you need to investigate this. So we're working with engineers to find these, to develop sensors that make sense. Yes, so that there is not a huge tower next to a beehive with, however, with a windmill for energy or something, but something small, something that can be integrated into the hive and doesn't cause too much trouble while beekeeping. We're working hard on finding new ways to do this. Another tool we use regularly is artificial insemination. We have in the past artificially inseminated honeybee queens on a regular basis. We have sometimes done several hundred over a week or so. We were involved in helping the beekeeping program, the breeding program of beekeepers in Perth, Western Australia, and helped them maintain their lineages by using artificial insemination. So here is the point where you can ask questions. Let me see. Are there any questions?
We don't have any in the chat yet, but if anybody else wants to unmute and ask, you can leave the opening here.
Okay. And you can hear me well, and you understand what I'm saying, okay?
Yeah, sounds great.
Okay, wonderful.
Oh, there we go. So one question came up. Abram, thanks. Is the Hive monitoring able to use video? Or what does it read in the hive?
Oh, that's a very good question. So at the moment, we are experimenting with hive temperature and with hive humidity and with gases like CO2. And the output at the moment is a line over the time where it's monitored. So you have like a line going up and down depending on the temperature during the day. What we would like to do though is to find out threshold values, and after those the beekeeper would receive a message saying, hey, the temperature in this hive is very low, maybe go check it, you know, maybe your hive is getting smaller. But we would actually like to do this so that the beekeeper gets a message before the hive gets too small to be treated or to be taken care of. So not just your hive is empty, that's a bit late, right? So we would like to find out before, like when it starts. And that's actually the tricky part, because when, for example, now when it was so hot, you know, how do you know whether a hive is going to overheat or not? So at what point during the day do you know? And you want to know as early as possible, right? You want to tell the beekeeper a couple hours ahead of time that the hive might overheat. So these are the discussions that we're having in the team. We're reading literature, we're finding optimal temperatures for hives, and we try to get those threshold values also together with the engineers, so that the message is not just a line going up and down to some peaks and you have to read that, but that there is actually a bing, you start having a problem. And then the camera part, we did look into that, and we will keep looking into that. Like, for example, having a camera in front of the hive to distinguish between bees that carry pollen or not. We have an engineer working on artificial intelligence, and he's been busy trying to also tell the camera and the work with teach the camera and the computer what's a worker and what's a drone. That was actually a project he was supposed to do this spring and summer, which he couldn't do now. So he will follow up on that. He will keep doing this. Did that answer the question?
Yeah, that's awesome. Almost like a facial recognition for the bees, huh?
Oh, that would be great, yes. Oh, that's Hilda. Oh, she's bringing in pollen from the whatever. That would be really nice, yes.
The fact that it could identify a drone versus a worker, if there's pollen or not, like, that's pretty amazing.
Yeah.
Has there been, to follow up on some of those questions with the monitor, has there been any research done with like sound or the pitch of a hum or anything like that?
That's actually a very good idea, because the drones hum more deeply than the workers, and you can hear it when they're coming out, right? That's a very good observation. And we have been discussing with people from Italy that measure vibrations, and that would be interested to know whether those vibrations, you know, could be actually measured already at the entrance of the hive when the drones are humming, or whether we would use a microphone. I think the next time we will deploy sensors, the idea was to deploy microphones, and it's definitely worthwhile to distinguish drones and workers.
Super interesting. Thanks for that. And with the monitors, have you had any issues? You were talking about seeing if they build up. Have they built up propolis or start building comb or anything in those abscess spaces?
It actually worked pretty well, as long as you keep the bee space free, like that image of the frame with the mosquito netting around the monitor. It actually worked quite well. I was quite impressed, I have to say. There is another monitor we have in Hives right now that is a bit bigger and a bit more clumsy, that just measures temperatures and humidity, and we have that in a plastic. And I don't know how the bees, I have to ask Jamie, the beekeeper, whether the bees have started building around it, but it worked pretty well last time. So as long as you keep the bee space free between the frames, it seems to work. But I mean, of course, I'm sure that once we have the really expensive monitors in there, they'll start building. I mean, that's bees, right?
Awesome. Thanks for that. There's a couple other questions I think they might wait till towards the end, but if you want to just continue on, I think you're talking about the facilities next. Some of the questions are regarding the facilities.
Okay, I'll talk about those and then we answer the questions that are still left.
Awesome.
Let's see. So these, let me see, I have to make the thumbnail smaller. Here we go. So we've just visited the entomology building with the lab and the researchers. And so let's walk over to the teaching yard. That's about a 5-minute walk, 10-minute walk. Here, we are at the teaching yard. That's a bee yard right on campus, here. And we have our 20 up to 25 hives that are very nice, that are big and friendly. And we keep these hives here for the students who come and learn bees and beekeeping with us, because of course, when you learn bees and beekeeping, you want to work on friendly hives. And here are the students hard at work. That's the first time these 32 students open a hive. That's always a very exciting moment. And I'm always thrilled to see them when their suits fit, their boots fit, and 2 students per hive, and they open the hives and get excited. The first time they open a hive, they're a bit reluctant because of course bees can be scary. And even the nicest bees, you know, you'll have the odd one flying or whatever, right? So, but after a couple times at the hives, they get so much more confidence. And I find that always very, very exciting to work with the students in the bee yard. The students give very good feedback, and we have a waiting list of like 68 students for 30 spots every time. It's a very successful course. We could teach it a lot of times. We teach it once in spring and once in autumn. And we went from class size 20 to class size 32. So we had to do some adjustments. This is our trusted bee truck, Melly. She's one of the oldest vehicles on campus, but she still runs, and that's good. She has had a couple nails in her tires here from AgOps, Agricultural Operations, where she stands right now. And but she's a trusty bee truck for transporting water and hive parts and whatnot. So yeah, she gets fixed up by the garage people regularly. And you can see we need her now to go from the teaching yard here to the holding yard. And this area here is called Agricultural Operations. That's actually used to be the citrus research station, and that's the core of UC Riverside, because UC Riverside developed from a citrus research station. They keep a lot of rare citrus genetics in agricultural operations, so it's important for international citrus research. There's also turf research going on, and other crops are grown here, like avocados. This is our holding yard. And the holding yard reminds me a little bit of Australia because there are so many eucalypt trees, and it's nice and shady. And the bees that need to recover, they are here in the shade. Bees that are smaller nukes, they're a little bit back here. They're on hive stands, smaller hives or hives that we have here just to recover and produce some honey. And also hives they might not have the very, very best of tempers. And we will discuss that later, because this holding yard is the most isolated bee yard we have. We keep all in all about 80, between 60 and 80 hives, depending on how many we have split. But yes, this is the area that is shadiest, where hives recover, and where the hives with the iffy temper are stored. Within walking distance from the holding yard here, from this little forest, is the research yard. And the research yard looks like this. We have it fenced in, and we can assign groups of hives to people working on them. So if somebody works on sensors here, somebody else can work on reproduction here. These bushes will grow, hopefully, and then provide some shade, because this yard is rather sunny. And the cool thing is this yard has Wi-Fi. So to pilot with sensors before you know exactly how many data you send where, it is very good to have Wi-Fi, like you can have a sensor on Wi-Fi here in these hives, for example. And the good thing is also we have an indoor lab right next to the research apiary. And you see Jamie here grafting. He's grafting new queens in the research indoor lab. And the indoor lab also keeps here, we can keep hives indoors. We've just started doing this with 2 nooks to test the situation. We have the exit here with a hose, and Jamie's experimenting with new clamps to clamp them off. At the moment, we still have had some issues with ants, but I think he's gotten that under control as well, because especially with younger hives, we really do have ant issues. This is our storage barn, and this is very handy, of course, to keep all the boxes and whatnot. And we have 2 freezers here, a chest freezer and another one. When we have a colony that dies, we freeze the frames before we use them again. And so we have 2 freezers where we can do that. And then in the next step, we can put frames that need to be stored cold in a cool room. So we have a walk-in fridge, which is absolutely fantastic for us. We had some issues with moisture in here. So some of the pollen frames got moldy, but we have a desiccator in here now that works, and it's gotten much better. So we don't always have a slush of sugar water and honey down here. You can see some honey frames here. We store our honey frames here, either those for human consumptions, but also those that we give back to the bees in winter when we have them in deeps, if they're extra frames. So, and the honey from this fridge is extracted and goes to our Bees cafe. This is a bee cafe, a honey cafe on campus at UC Riverside, and Dining Services has a memorandum of understanding with Cyber, and part of the money that they make with our honey goes back to the salary of our beekeeper, which is of course fantastic. And the food here is infused with honey, so there is salad sauce with honey, and there are coffees with honey that we really like. It's a nice place to hang out, close to the engineering department. So this is another point where we can ask questions if you like.
Yeah, does I guess the food preparation, do they take all of your honey? Do you also sell it to the public outside of storing it and back to the bees? What else do you do with it?
Well, actually, Bees basically they would use all of our honey. So we keep a little bit back so the students can spin it out. We have 2 times 32 students every year. So they get to spin honey and take some home. So that's part of it. We have a honey tasting at the insect fair where we give some of the honey. We keep some honey for gifts so that we can have a sticker with our logo on it. We had a bit of, oh yes, we had some candles. We made some candles with the wax cappings, and candles were given away as giveaways by the communications department at UC Riverside to advertise that we're here. So it's kind of like that. We are, because we are at the university, we're not allowed to make a profit. because we cannot be in competition with anybody who does commercial beekeeping or who sells candles or wax or whatnot. And it is also not our job to produce candles or honey, right? It's our job. It's a byproduct of our research because we're research scientists primarily. So this way we can basically wash the honey money and get something back into our funds, which is good, right? Because we would be expensive employees if we made candles or honey, because yeah. So yeah, any more questions?
Yeah, Avram had another good question. Talking about the 3-mile rule. So when you're moving bees between the yards, are you paying attention to how far they're going? And can you explain that a little bit?
Yes, well, so actually when we move bees from the, we cannot make 100% sure that the bees wouldn't move between the holding yard and the teaching yard. But so we do move them, and we don't see, like, I will talk about the, okay, let's see. We have some drift, like in the teaching yard, when we have the hives all in one row, it is easily possible that we have in tendency the hive at the beginning and at the end of the rows, that they have a bit more bees than the ones in the middle of the row. But because, and so that is something that we should be addressing actually. And also we should probably start looking into painting the entrances of the hives. Back in Australia, each hive had a pattern and they all had colors. Here they're still white. And we had in mind to make a big thing on campus, a little party on campus where people can actually paint our hive boxes, maybe for a little fee, just for fun. And then we take some pictures of the hives and maybe make a little competition or so. And that would make it easier for the bees to recognize who lives where. I cannot exclude that we have drift. No, but we do keep the hive tools clean. So we clean them in the smoker between the yards. And as I said, we freeze the frames before we exchange them between yards. Sometimes in the same yard, you know, when you have a weak colony and one that is like overflowing with honey, we might feed a honey frame to a weaker colony or some pollen or so. So within the yard, we will have drift, but we monitor mites regularly and have an eye on that so that we don't transfer too many parasites. Or I mean, we try to avoid it as far as possible. In Perth, we had a barrier system. So the hives had certain blocks of hives were assigned to people. The orange hives were the ones where I could do my research in, and the yellow ones were the ones that somebody else could do research in, but I was not allowed in there. And in between manipulating hives, we would really thoroughly clean the hive tools. But we only had one yard there, so we had to do it by color block.
Very interesting. Thank you. Talking a little bit about the management of the hives in Riverside, somebody just asking, you know, with the climate in Riverside, when do you experience dearth? When do you start feeding the bees back that honey or additional syrup?
That's actually a good question. So I can kind of, maybe let me talk about this with the management. We have a level of how we assess how much honey and pollen is in the hive. And according to that, we start feeding. Coming from Perth, I was in the beginning kind of against feeding because we hardly ever did that there, because the beekeeper had a different method. She brought the hives over winter into an area that was warmer and more, and where there were more flowers. She just moved our hives. We had 50 hives there and she moved them. And the ones that were moved, they came back in spring and were ready for research. I was also less involved in beekeeping there than here. So I learned that in this climate, you definitely have to feed them. And Jamie has been working very hard on keeping them at a high level. So he feeds them sugar water and pollen shakes. And when a hive is not strong enough to, when we think that they don't collect enough food by themselves, and we have a bit of a level of how we judge that, then he will start feeding them so that they can come up to a certain level where they can take care of themselves. So shall I continue with that hive management then, or are there more questions?
Yeah, that sounds like it'd be appropriate. Yeah, let's hear the next section.
Okay, I'll do that then. Let me see, let me minimize this. Okay, so we have regular hive maintenance tasks like everybody else. One of them is, of course, to keep the hives supplied with water. We use these watering stations that Jamie has devised that have a vacuum basically where the water goes up to the rim here, and there are pebbles and there are cleverly placed holes that can be plugged and or open depending on whether you feel water here or not. So it's basically a vacuum and the water just comes here up to this level. I find these stations quite clever, but they have to be steady, like it has to be flat, otherwise they overflow and you have an issue. But they're well used, especially in the teaching yard where it's quite dry. And then we have ant issues that we have to keep an eye on. This was one Monday morning in the lab, in the field lab. The bin had been sitting here and the ants just made these nice little frames around them. And of course we have the ants at the hives as well. That's why our hives are on stands, and the stands stand in containers that contain water and oil to keep the ants out. We had some rodents eating away on yogurt containers, and so that didn't help much. So now we're going over to cans to keep the water and the oil in cans, so the rodents cannot eat the cans. And now to recording hive data. When we go out to the bees, we have these clipboards and we have little fields where we can fill in information. The information we fill in is about the colony size, the mite counts, the status of the queen, how defensive a hive is, and how productive a hive is. I'll give you some examples. One measure of colony size is the number of frames that contain bees on both sides. So when we look at this here, there is at least 1, 2, 3, 4 frames where bees are visible on both sides, and then maybe half of one. If you want to be really precise, you can go underneath and have a look at the box again, and you see again 1, 2, 3, 4, maybe 4.5. So yeah, when you look at this, it's between 4 and 4.5. If you're optimistic, 4.5 frames of bees for this colony. So that's kind of an over the thumb or back of the envelope calculation for hive size. We do a similar thing with brood frames as well to give an indication of colony size each time we check a colony. That's about every 3 weeks. At that point, or a little less frequently, depending a bit on the weather and the size of the colony, we do mite checks. For mite checks, we use 2 methods, the sugar shake and the alcohol. For the sugar shake and the alcohol, we use this piece of metal. It is very convenient. You can shake the brood frame over this piece of metal, pour the bees into this little cup that contains between 300 and 400 bees. You pour this into the sugar, you shake the bees, and you count the mites. If we see more than 3 mites per 100 bees, if we calculate more than 3 mites per 100 bees, we will treat depending on the weather. We use thymol or formic. And the same with alcohol, but the bees will die in alcohol, of course. So you have to be especially careful with the queen, like have one more eye out, make really, really sure you see her, of course. And then you can keep the bees in the alcohol though, before pouring them here, counting the mites and the bees. And you get more precise measures that way. And also somebody else can count the mites and the bees. You don't have to do that at the same time as you'd go through the hives. So for weaker hives that shouldn't lose 300 bees, we use the sugar method. And if we're in a hurry and the hives are strong, we use the alcohol method. We monitor the queen status. And you can see we use these pens for the queens. And what we monitor is whether a queen is the purchased one that we had originally in the hive, or whether she's a self-raised one. To do this, here we have a nice graph. I'll walk you through this. So this is about mites and the queen status. And you see here on this axis the number of, oops, I'm sorry, I have to go back here. Here we go. I shouldn't talk with my hands while holding the mouse. So you have here the number of mites per 100 bees. You have here whether the queens are purchased or self-raised. And you can see here that in case of the purchased queens, the colonies from purchased queens in tendency had more mites than the queens that raised colonies, the self-raised queens. So colonies from self-raised queens had fewer mites than those that were purchased. So we needed to treat colonies from self-raised queens less often than those from purchased queens. And this is exciting to see. And we do treat all the colonies, no matter what, whether they're purchased or not. If they have mites, they're treated. But it seems that we didn't have to treat the self-raised ones as often. These data are from the 60 or 70 hives that we kept between January and July this year. So this is the newest data on our hives. The queen status is also marked according to the year. This is a convention, like the purchased queens we mark according to whatever color is assigned to queens that year. So this year it's blue. So purchased queens from this year are blue. The ones from last year green. And we have like 2 or 3 left from 2018, which is in itself nice to see that some queens that are purchased are able to survive that long. The same is true for self-raised queens. If we have about 2 or so left that are self-raised, and it's actually from these groups of queens that Jamie has been grafting, because that the fact that they survive Riverside weather for that long is a good thing. And then when they're from 2019, they got a yellow dot, and this year's got a hot pink one, because I like pink. Let's go with here. We mark the lids as well, according to queen status. Purchased queens get a 3-digit number, self-raised queens get changed to a 4-digit number, and we mark down, okay, here this is a purchased queen, she's blue, that's what we expect in there. Okay, here we expect a self-raised queen, she's pink. If we see anybody else, like if all of a sudden we have an unmarked queen, of course we know, okay, they've been requeening again, and then we will report that. And you can also see, we write down, okay, this hive was treated with formic in May. Now to defensiveness. Of course, when you have self-raised queens, you will have issues with defensiveness if you're not careful. And we have the number 1. Those are our famous teaching hives. They hardly react when you open the hive. 1.5, they get slightly agitated. They start, humming a little differently. The 2 number means the guard bees start flying up. 2.5 means the guard bees start bouncing off your veil. And number 3 would be they start stinging and following you. Sometimes these go hand in hand. Maybe you start at this level and then you end up on this. But this will be an issue for us. So we'll have a very close eye on the ones that start bouncing off your veil. If they do that 2 or 3 times in a row during an inspection, they'll be moved to the eucalypt forest and split and requeened. And this is not on at all. I mean, these guys, they get split immediately and requeened. And as a result, we can say, again, here you say the similar graph to before, but this time about defensiveness and the queen status. You see here the average defensiveness between purchased and self-raised queens. And you can see that the defensiveness is virtually, it's like basically the same. So they're between, barely caring and going a bit zzz. So it's actually quite nice to see that we are able to contain the average defensiveness is good, and we keep the hives in one box. When they get bigger, we might have issues with higher defensiveness. This is something we can do research on eventually. But again, between January and July, keeping the bees that we do the way we do at the moment. And I mean, it's Jamie who does that work mostly. He does a really good job in keeping defensiveness down. So we've talked about colony size, mite counts, queen status, and defensiveness. Does anybody have a question?
There are. You were talking about mite counting. Have you ever done comparisons? Because I know there's always debate, right? Sugar shake versus alcohol. Have you done comparisons to see accuracy in those different mite tests?
Yes, I think we have, and they are not far apart. So, oh boy, I don't know by heart how accurate they are and whether I have actual proper data, but we've done, I mean, I know at least that, as you call it, back of the envelope, it's quite okay. It's quite, I think it's more of a speed thing. We are quite good with that measuring cup. We kind of estimate, you know, whether it's 300 or 400 bees, and you get the treatment level. For the treatment level, it's okay to do the sugar shake. You don't need the alcohol. As long as you treat when the hive reaches 3 mites or more per 100 bees, per estimate of 100 bees, you're okay. The alcohol, like when you have 80 hives and you have people who help you, you can be quite quick going through the hives, just doing the mite checks one after the other. And then you don't have to every time, you know, shake and count and whatnot. You just alcohol, 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 and you get it done. Like you have a quicker way of getting it done. And when the hives are strong enough, you just look for the queen, do the alcohol shake, and somebody else takes care of the counting and the mites. So that's kind of where I see the advantage of the alcohol versus the sugar. And then maybe, okay, if you're on a very precise project where you need to know exactly, you know, how many mites and how many sub-mites and what age or what status, then you would probably do the alcohol test because you have to distinguish between different mite stages or what have you. But that's a different thing. I think for every day, I would say the sugar works very well, actually.
Ultimately, if you're at 6 or 9 mites at that point, then you're going to be treating anyway. It doesn't have to necessarily be exactly 300 bees. I know one for the UC, the Master Beekeeper program, they promote that whichever route you go, to do it consistent, right? So if you're doing sugar shake, you're going to continue using that same method, right? So at least at that point, however, you know, effective or not that is, it's going to be consistent from test to test.
Okay, yeah. I mean, I guess if you have, it's a good argument. You could say, I guess if you don't have that many hives, you could just go with one method. But I think they're as I said, I think they're rather comparable. And if you just go, you want to know whether the mites go up or down, kind of for what we need. For what we need, it's kind of okay. And we don't want to, you know, the alcohol is just quicker and the sugar takes a bit longer. And if we have a weak hive, we don't want to use the alcohol. So kind of, it seems okay to do it that way. I would have to ask an exact comparison or so, but it's relatively rough, right? The measure. And there is actually something that we would love to measure, which is larvae when they are in the neighborhood. So honeybee larvae that are 6 days old are very attractive to mites. And but when they only, so there have been experiments, Boris has done experiments like that with a postdoc in China. I mean, the postdoc did the experiments in China. And the larvae react with an alarm smell when they are in touch, when they see a mite coming, or I mean, when they just feel the mite crawling on top of the larvae, before the mite even bites. So the larvae will already emit a smell to alert the workers that the larvae has a mite on it. And if we would find a sensor that would measure this alarm, this smell, we could quite accurately predict whether how infected the hive is. That would be awesome. And I mean, the idea is cool, but now we have chemical engineers trying to find, you know, to isolate the smell and to make sensors for it. I mean, we know the name of the smell, but they try to train a sensor to detect the smell, and then you have to find the smell in the background noise of the smells of a hive in general. So these are the struggles that we're struggling with, right? To refine that method. That would be just awesome. And then you would get a ping on your phone. Hey, mite levels in this hive are too high, treat. So, you know, that's where we're going, right? That's the Nobel Prize later on.
That is amazing. Are you, when you're tracking those numbers, are you using Excel? I know with so many hives, right, you might have a more apiary level like app, or what are you using for
No,
that?
not yet, but that's a good thing. So one idea was to put this to put this paper, to not use pen and pencil to fill in this paper and then copy it down into a Google spreadsheet. We use a Google Sheet and share that, which is just so boring, right? You sit there and you enter the data, and then you get cross-eyed by entering the data for hours on end, and they all look the same. So it would be awesome to do this on a on an iPad, actually, right, or whatever tablet. But the thing is, you're wearing gloves, right? So there are gloves that have some contact. So one of our engineers has sourced gloves that have a pad with which you can use the phone. But my gloves always get so goopy. I don't know. Maybe use a little stylo. But when you use that, then you have an additional tool you can lose in the bee yard, right? So, so it's not been, we haven't found the optimal way, but if anybody knows or has ideas of how to use a tablet safely in a bee yard, that would, I mean, that's something that would be cool. At least in the one with the Wi-Fi, we could try and enter it directly into the Google Sheet instead of copying it down, right?
Yeah, it's all great stuff. And as backyard beekeepers, most of the club, right, we're talking about you know, maybe one or a small handful of hives, right? And there's a lot of different commercial apps out there, and but I'm sure with so many, even if you were using an app, that would be kind of a different application.
I'd probably have to check those apps. I haven't really looked into that. Maybe we should have a look at that. Yeah, maybe you can send me some links or so and check them out, because yeah, we've been basically reinventing the wheel, maybe.
Yeah, being able to share that data, I'm not sure which apps have been best for that, but awesome. Thanks for that. Yeah, nitrile gloves, I know, can usually touch a little bit better. I always use leather because I hate getting stung.
Yeah, no, I don't like it either. And then when the gloves get thin, the stings break off, and then you have the stings in your hand for ages. Oh well.
Not fun.
Nope.
I think there were a couple other, maybe towards the end. I'm not sure if you have other actual presentation.
Yeah, I have some information on productivity, but it's up to you. It's 8 o'clock. I've been I've been longer than I thought I would be, actually.
Okay, well, it's all great information. We usually, like in a live meeting, we might take a quick break and that kind of thing. But if you want to go ahead and keep moving on, and people can always grab a coffee or something if
you wanna how is everybody feeling? Is anybody snoring yet? Not yet.
I'm getting thumbs up and no shakes. Yeah,
okay, good. It's not much longer, and I didn't think it would take that long. I've practiced this talk, and I was through in half an hour. So it's usually what it goes, when you have. But yeah, okay.
You're good. Let's keep going.
Okay. Let's go on. There. We estimate the amount of pollen and nectar in our hives from 0 to 3. 0 is none. 1 means they started collecting some. 2 means they have enough stores for the colony. We don't have to worry about it. And 3 means they have enough to share. So if a colony is weaker, we can take some from here and give it to a weaker colony. And this was the question, right? When do we start feeding? So if we see that there is none, or if they just started collecting and the colony looks weak, or the weather is not getting much better, like if the starting of collection is not due to weather, but because the colony looks a bit weak, then Jamie will feed them. And you can see that when you look at the pollen per bee yard, you see the mean pollen stores here. So between, we have enough for the hive, or we can share some, and you compare the different yards. The holding yard, that's where the eucalypt forest is, the teaching yard where we have our students, and then the research yard, just a couple steps away from the holding yard. And you can see that if you look at the standard area here, that these are actually different. The holding yard, in the holding yard, the bees collect more pollen than they do in the research yard, even though these two yards are only a couple steps away from each other. And in the teaching yard, they're somehow in between. I really don't know why, because if I was a bee, well, no, apparently not. If I was a bee, I would know why. But this is just a couple steps apart. And I really don't know why the bees collect less food in the research yard than in the holding yard. And we have the same picture for the nectar. So we have the mean nectar stores here, and we compare them between the holding, the teaching, and the research yard. And you can see again that the holding yard, that we have more than in the research yard, and in the teaching yard, we have somehow in the middle of the amount available. And then we come to the drones. So if you translate productivity into the production of drones, because drones are always hungry, and you have to be a strong colony in order to be able to maintain these young little drones. I always find them the cutest when they're newly hatched. How do you translate that into drones? So are drones here or not? It's a very easy thing to measure. Either you see them or you don't. And when you look at this and you compare again the holding yard, the research yard, and the teaching yard, in the holding yard, about 60% of the colonies had drones present when we were checking on them, 40% in the teaching yard, and fewer in the research yard. And it's really, again, a question why. So one reason we came up with that might be the shade in the holding yard, right? So that bees who don't have to constantly fight to have a temperature that is suitable in a hive, they don't have to cool their hive constantly with water, they have more energy to go out and fly and collect pollen and nectar. Maybe that's the reason, and that's why we started now newly placing monitors, just temperature monitors into the hives and testing different lids. And that will be a story for another time to see how the differences in temperature are between different types of lid and insulation. So that's the last part of my talk. I thank you for listening and ask whether you have any questions.
That's awesome. Thanks again, Barbara. But when we're all done, I'll let Liz go ahead and close up. But there were a couple of questions that kind of came in and out. There was, when you were talking about the different queens and a couple queens lasting, since 2018, obviously you're carrying some queens over. Is there a certain indicator or something when you decide in the different yards to requeen?
When we don't find eggs anymore, or when we find, well, we sometimes let the queens requeen themselves. And when we have drones in the yard, we let them requeen, right? But if they requeen and we don't have drones, and then we see them requeen again and again and kind of getting weaker and angrier and so on, then we order queens in. So we have, we requeen, what do we do, twice this year? Or in 2 rounds, like, let's say that, because I don't know exactly, which ones were requeened again or not, but in tendency between once and twice a year at least, but they self-requeen quite often. The first round of new queens we did didn't take very well, and we had a lot of self-raised queens afterwards, and some of them didn't take very well either, so we bought a whole lot of new queens and tried it again. That's why we are so eager to somehow find out more, to get into bee breeding, to get into queen breeding and raise our own queens.
Yeah, it sounds like an amazing project. When you're inspecting those hives and you're keeping data on mites, for example, are you also keeping data on the individual frames within the hives or in the individual boxes?
Well, so that's something that we were discussing quite a lot about, how we label the queens and how we label the boxes. And the equipment it's interesting to know how the equipment is doing. We have worked on differences. I mean, I could show you data on, the queens and the yards come together and the combinations, but because I don't really know why, I cannot really explain it. I didn't want to go too deep into that. But in short, we don't keep track of, well, we label the frames with the year we put them in. When they were new, they get a year. But we should actually get a brand probably and brand each frame and brand the boxes, because we have a cycle of, cycling them out. So we inherited a lot of equipment when we came. Yeah? there was just some, anyway, we inherited a lot of equipment and some of it was more usable than others. And I'm still working on trying to find out whether the frame is usable or not. One criterion is, do the bees rob from it or not? So if you leave the frame that is empty out or that just has a little food in it, if the bees go rob from it, then it's probably still usable. But if the bees don't rob from it, just don't use it anymore because they don't like it. I mean, it's kind of a crude thing, but we do try to rotate frames out. So when they get too dark, or have you noticed, by the way, how like the wax is usually light, right? And then it gets darker, but I haven't figured out fully how long it takes until a frame is fully black and how long it's still usable, you know, until which darkness. Do you have criteria for that?
You might have 15 different opinions coming out. So I'm going to hold back that feedback because there's a couple other questions.
Right.
I personally don't. I have several people in the message box just saying thank you very much. And there's a couple different languages being posted. And I think they all say thank you.
Herzlichen Dank.
Oh, bitte, gern geschehen.
Ja, bitte, bitte.
That's awesome. Yeah, so thanks again. There's a couple other, I know Chris had some questions about yourself. Do you have hives yourself? I'm sure the work that you do at the campus is quite exhausting. Do you have other hives at your home or anything like that?
No, I don't. Actually, we have a beekeeper who does most of the work, and I am actually honored to help out occasionally, especially now. For last year, I was in charge of the hives for 9 months because our beekeeper was on sick leave before we got a new one. And so I was far more involved than I used to be. And that was when we had help, like Mark helped and some other beekeepers helped out and taught us to trade for here, Southern California. We came from Australia, so things were a bit different. So we had to learn how to treat mites and how to do the whole season in California. It was a big learning curve, but I'm glad I got my hands into beehives here and got to find out a bit more, you know, not from a desk, but actually more practical side on how it's done and how hard it is here actually compared to Australia where there are no mites, right?
Yeah, we do have some challenges here. Thanks for that. Couple suggestions that came across using a stylus for iPads. Somebody mentioned maybe using the back of your knuckles with the glove.
Oh, okay.
I haven't been able to do. Thanks for that, Misty. Abrams mentioned wrapping the iPad in plastic wrap.
Yeah, that might work. Mm-hmm.
I'm gonna test that too. Mm-hmm. More thank yous. A couple questions just asking if does UC Riverside ever do? Maybe we could even do a club tour or something of your grounds there.
Oh, definitely, we would love to do that, and we would love to have people over. We do tours, yes. So we would love to do that for you guys, and I hope this will happen at one point, right? So yes, and we are also, we're open to that.
That's awesome. So maybe when the campus opens back up, right? Of course, UCR is shut down right now, like everywhere else. Yep. And when we open back up, that'd be great to arrange for the club. Thank you for that.
No problem.
I think that covered all the questions. Liz, I don't know what your timeline was for tonight. I'll let you take over.
Yeah, I just had a few things. I wanted to know how the grant, if you heard anything back from the grant writing that you had done a few months back.
Yes, so I sent an email out to a couple people about that. So there were 2 grants that we applied for. One was a letter of intent for a conference that we want to hold the end of next year. Right. And we got a reply back for that letter of intent saying we should apply for we should write the full proposal and apply for the money. The other one was a pre-proposal for the breeding program, and that was not successful. So we did not get selected. They said the program was basically too ambitious, and they liked the team and that they really mentioned the letters of support from everybody. And I thank everyone who helped with that. I think that was worth a lot. So it got us noticed that the cooperation between the beekeepers and us seemed to be good, but they thought the project was too expensive and too ambitious. So what we are working on now is basically more proof of concept, right? Evaluating more data like the ones I showed you, but going a bit more in detail and then trying it again somewhere else or there.
And so I did want to mention that we had talked about this before as far as using our beekeepers, you know, in our clubs and everything. So if anyone is interested in any type of research project with Barbara and UC Riverside and what they're doing out there, you do have to be a current member of the OCBA in good standing, so that we can have good communication with everybody. But if Barbara or Boris, they have something that they want to test or research, by all means, we will participate. And so anything that you need, please let us know. As you know, Orange County, we have members that are in Riverside, Orange County, LA County, so there's a wide variety. And all the areas that you've learned from now, they can vary drastically just within a couple of miles, you know, as far as the environment. So yeah, keep us in mind for that. But yeah, as long as they're members in good standing with the OCBA, they would be able to participate in that. So did anybody else have any more questions for Barbara? After COVID is done and all this is done, we will be definitely setting up tours and different programs again, getting to meet with everybody, you know, back to that social in-person meetings. But if anybody else has any more questions, please get them in the chat ASAP, and Rick will go over them. And I want to thank everybody for coming on tonight. It's been a little break. I will also be sending out the Hangout message for the middle of September, the Hangout chat that we do. And also I'll be sending out the memo for the APIS-M chat thing that will be going on next Saturday. So any more questions on there, Rick?
There was one additional one in regards to the trees and planting that go on. Do they at UC, do they spray any of the orchards?
That's a good question. We've been in contact with the people in agricultural operations. They do spray. We have received some messages that they spray, but we don't. We are quite sure that we do not receive every message whenever they spray. I think they only tell us if it's very close to one of our yards. So they do spray. We do have, but I do not know exactly what they spray. And it's definitely worth getting more information, because we have we probably have quite some issues that we're not even aware of with the whole spray issue.
When you know that it happens, do you cover the hives, or do you take any measures to help protect the bees?
Not at the moment. It's not at the moment. So because usually they tell us relatively short notice, and it's usually not just right around the corner, and it's not so easy to find out where the bees are exactly foraging. So one thing we do is, as I said, we have these watering stations, because the bees could drink from the bottom of the trees where some of the pesticides will probably be administered through the water, you know, where the soil is moist, and the bees might like that. But there are other areas where the bees can drink from water that might be contaminated. So we try to provide them with clean water at least. We do see occasional piles of dead bees in front of hives, but we don't really know whether this is from robbing or whether this is from you know, attempts to pirate them from hives that live in the environment, not you know, survivor hives somewhere in the environment. So we probably do have issues with spraying, but we don't really know how much.
Gotcha. I think that was the last question that I'm seeing. Oh, there was one more. When you guys are doing either self-raised queens, or even when you're doing or doing insemination, where are the drones coming from? From your own yards?
Yes. So we have when we want to have drones, we put drone comb into the colonies, and they raise those. We have drone comb drawn out now, so it took a while until the plastic foundation was drawn out, but now we do have it ready. So whenever we want drones and we feel a hive is strong enough, we will feed them. Jamie has an awesome pollen and sugar shake, like really protein shake on top of them. And that will cheat them into thinking it's spring, even if it's not. And they will hopefully attempt to produce drones. We have quite a few hives producing drones quite late into, so like we have some that are still producing drones or were at least 2 weeks ago.
Are you isolated enough there in Riverside? You don't have to worry about outside potentially Africanized bees coming in?
We do have to worry about that. Well, worry. The drones might be, some of them might be Africanized, but even though they are self-raised, those queens, even though they do for almost 100% have some Africanized genes in there, the temper seems to be bearable. I mean, we had some wild hives. We just had to really split them, and we try to keep them to one box at the moment. In the beginning, like in 2019, I let them grow to bigger, like 2 boxes, and then on top a honey super and whatnot, and they became almost unmanageable. It became so heavy. So at the moment, we keep most of our hives to 1 box. And then if they seem to be able to have the workload, the capacity to do the work, we give them work to do, like either frames that are moldy to clean up, or honey supers to build up, or 2 honey supers or so, depending on how strong they are, how vigorous they are. We keep them to 1 box. We take out brood and share it with weaker hives, and we pile on work for them to do to keep them busy.
Super interesting. Thanks for that.
Yeah.
That's all the questions we have in the chat.
Okay. Well, thank you so much for having me.
Thank you so much, Barbara. It was great.
Okay, I'm glad you liked it. I had fun.
We'll have to do it again.
Yes, I'm looking forward to doing it live once too.
Yeah, I think everybody is.
Yeah, but yeah, thanks so much for all your questions and the interest. And yeah, I'm looking forward to keeping in touch and working together. I would like to do a bit of a workshop on the breeding program, actually. So if anybody of you is interested, just get in touch via Karina or sending me an email or something, and we can discuss this whole breeding issue. I haven't gotten that many candidates yet.
Well, if you go ahead and send it to me, I will send it out to, the membership.
Yep.
And I'm sure it's not because they don't want to, it's because they don't know about it.
Yeah.
We have a lot of people that are more into the scientific and the research side and want to be involved with something like that.
Mm-hmm.
So yeah, send me an email on it, and I'll send it out in the Hangout, our next message, which should be going out within a week. And then, they can get directly in touch with you or Karine. Where are you, Karine? Wave, Karine.
Yeah.
We could do something and record it as well, and that way you could share it more broadly through a YouTube link or something in the future, if that'll help too.
Yeah. Well, at the moment it's fine. I've been keeping that information about the grant and the whole breeding program among the people who were working on this. But because I didn't get that much feedback, I thought maybe I go a little bit wider. So yeah, let's try. I will send, maybe I'll draft an email, and then this can be sent out, and we'll see what comes back. We'll find a time, maybe with a when to meet or so from the people who are interested, and then we get together and see what comes out, how we go about this.
Sounds good. Thanks, Barbara.
Okay, cool.
All right, Liz, any last comments? There you go.
No, I think that's pretty much it for now. Everybody please be safe and stay cool, and protect yourselves. And I'll be sending out the information for the Hangout for the middle of September for APISM's chat next Saturday. And if I get something from Barbara before that one goes out, when I get it, you guys will get it. So thank you everyone for coming tonight.
Thank you.
Thank you. Have a good night, everyone.